small and in the white sides. So the material that we work with can come from sediment, it can come from air, rock, even water, or pollinator organisms, and so on. So there's a lot of different materials we are working with. And this material can then give us indications about where it comes from, which environment, for example, or which organisms it came from, or the conditions it has been through. Yeah, but to not dive too deep into palynology yet, I may maybe first talk about my own background. Let me see how this works here. So I myself, I come from the Collaborative Research Center, the CRC 990, also called the EFFORTS Project. And the EFFORT project in long is basically studying ecological and socio-economic functions and trade-offs of tropical lowland rainforests and also transformation systems in Sumatra. So we aim at providing science-based knowledge on the one hand, how to protect and enhance the ecological functions of tropical forests, as well as agricultural landscapes, while also improving human welfare. So furthermore, this program aims to provide a baseline of knowledge and information on how to integrate agricultural land use and conservation issues. We are working in 4 major transformation systems. Here, rainforest, oil palm plantations, rubber plantations, and jungle rubber plantations. The Collaborative Research Center, as the name says, is a collaboration. So actually, we are working in 23 different projects with more than 160 researchers and staff working together. So the collaboration here is between 3 universities in Indonesia. I think Bahesti mentioned this already. The University of Bogor, the University of Tadulako, and the University of Jambi. And this is collaborating with my university, the University of Göttingen in Germany, through the DFG, which is the German Research Foundation. Of course, this project is quite big, so I don't work the entire project. I work in one small subgroup, which is subgroup AO1, studying long-term vegetation dynamics, plant phenology, and plant pollinator interactions in rainforest and transformation systems. Here, we basically work with 3 working packages. On the one hand, we have paleoecological studies, which is studying basically vegetation history, but also modern pollen rain studies. So modern pollen precipitation, everything that is precipitating from the air, and plant pollinator interactions. So how we can see the signal in pollen from vegetation on pollinator organisms. But more to that later. The principal investigator of this project is Professor Hermann Behling from the University of Göttingen. But we are also working together with our Indonesian counterparts, which is Professor Subiandi from the University of Bogor, Dr. Asmadi Saad from University of Jambi, Dr. Rika Rafiundin, and Dr. Nina Juita from the University of Bogor as well. Then there's myself. I'm the doctoral researcher, so I'm actually doing my PhD right now, and I am just a researcher in the project, and the associated doctoral researcher, which is Chung Nguyen, who also does his PhD in this project. About working package 1, we mostly hope to study the sensitivity and resilience of different ecosystems towards climate, fire, as well as human impact in the past. So as I mentioned, vegetation history. So we are trying to find out what vegetation we had in the past, how it looked like, and how it transformed through time and space. In working package 2, We study the relationships between pollen and vegetation, and also the influence of transformation and climatic oscillations on this pollen, how it is dispersed, how diverse it is. Yeah, because it is after all a genetical dispersal unit and an important keystone in ecology. In working package 3, we mostly study plant pollinator interactions. This is more collaboration with other projects. We just do it as a side project, let's say, studying also the effect of transformation on pollination services. And we work together with a lot of the other sub-projects of this big collaboration. Below just some examples. So in our working package 1, we study basically not only just the pure pollen, but sediment cores. So sedimental pollen from different sites. Here you can see the province of Jambi, and you can see these yellow dots, which are actually our sediment core from working package 1, in which we study vegetation history. And you can see they are distributed alongside an elevational gradient so that we can actually see how the landscape transformed through time in different ecosystems. Then we also have study sites from the CIC, which are our core plots. You can see them as these little squares here in red and blue from 2 different sub-projects. On the one hand, the core plots and the landscape assessment plots. However, I myself certainly don't come from Jambi, but I actually come from the University of Göttingen, the Department of Palynology and Climate Dynamics. This is where I'm working at every day, not right now, certainly because I'm still positive on Corona, but usually you will find me here. So we are in the center of Germany, in the northern center. Actually, we already had some snow the past days. And this is our department. It's located in the city center, nicely in the botanical garden. And you can see already that the leaves here are turning orange. And so this is autumn. And this is probably the state right now, winter, where all the leaves have fallen already. So here again, the AO1 team. So you actually also see some faces. So our principal investigator, Professor Beling, myself, Chung, and our counterparts from Indonesia. So let's get back.
Maybe before continue, I will try to translate to my student.
Of course, yeah.
Jadi di sini saya menyatakan bahwa yang akan kita pelajari di sini adalah tentang pengenalan terhadap palynologi ya. Kalau kita lihat di slide pertama tadi, palynologi itu berasal dari kata bahasa Yunani, itu artinya adalah sprinkle itu artinya butiran atau debu, atau sederhananya itu adalah ilmu tentang debu, tapi tidak sesederhana itu katanya tadi ya. Kemudian saya juga merupakan bagian dari Collaborative Research Center CRC 990, di mana beliau termasuk ke dalam grup A01. Di grup A01 ini ada peneliti utamanya yang tadi itu namanya Profesor Herman Dahling. Dan di sini ada 3 topik yang dibahas. Yang pertama itu adalah tentang paleoekologi, ya, jadi ekologi masa lalu. Kemudian di sini juga dibahas tentang modern polen. Jadi polen itu kalau kita bisa lihat nanti itu adalah serbuk sari ya. Kemudian ada lagi yang dibahas itu interaksi antara polen dan polinator. Jadi polinatornya ini bisa kupu-kupu, bisa lebah, dan bisa banyak sekali gitu. Nah, kemudian untuk wilayah penelitian dari A01 ini di daerah Jambi, kalau kita lihat peta yang tadi, sorry Safia, could we move to the map of your research? Previous one, next, this one. Nah, di sini ini objek tempat penelitiannya itu mulai dari kelinci sampai ke tungkal ya. Jadi di sini kita bisa melihat titik-titik bulatan warna kuning atau kotak-kotak itu menunjukkan tempat pengambilan sampelnya. Dan kalau kita lihat yang berwarna hitam itu adalah lahan gambut. Kalau misalnya yang berwarna hijau itu adalah daerah dataran rendah ya, dan yang berwarna agak tua itu adalah dataran tinggi. Jadi lingkup penelitian mereka itu mulai dari ketinggian yang paling tinggi sampai yang paling rendah. Jadi itu sudah dilakukan penelitian oleh tim A01 di CRC. Kemudian Svea ini bekerja di Departemen Palynology dan perubahan iklim ya, Universitas Kontingen, tapi karena beliau lagi dalam kondisi sakit jadi sekarang work from home, jadi tidak di tempat yang tadi. Dan timnya itu ada yang tadi disebutkan, ada Profesor Herman yang dari Göttingen, kemudian ada lagi Svea sendiri, kemudian ada Chung, kemudian ada Ibu Rika, ada Bapak Supiandi ya, ada Ibu Nina Juwita ini dari IPB, dan ada satu lagi dari dosen India namanya adalah Bapak Semadi Sahat. Okay, I think that's all. Okay.
Yeah, okay, thank you. Maybe you can also tell me when to stop and when you need to do some translations. Always get caught up.
Maybe we will translate every slide, maybe?
Yeah, I can make a small pause after every slide. Yes, sure. Okay, yeah. So let's get back to actual palynology. So what is palynology actually? First of all, maybe this word is quite new to you. So I will discuss this a bit. It's a branch of science dealing with microscopic and decay-resistant remains of certain plants, as well as sometimes even animals. And the pollen itself is the ideal microscopic proxy because, first of all seed plants, as you know, produce pollen. And the majority of this pollen has a pollen grain wall, which is highly resistant to decay. So it can prevail over millennia, meaning that if you actually study an environmental archive, for example, a sediment core, you will find pollen in there, which is already quite old. Then pollen grain walls have a wide range of morphological diagnostic attributes, which allow us to actually identify pollen to family level, sometimes genus level, and rarely even to the species level. Pollen itself is produced in the anthers, as you might know, and is basically a unit for pollination, which then pollinates the female parts of the flower, produces seed, and then causes germination and a new plant to grow, which then again produces flowers, which produces pollen. Okay, Hesti.
Jadi palynologi ini adalah cabang ilmu yang berhubungan dengan karakter-karakter yang kecil ya, mikroskopik gitu. Jadi untuk palynologi ini, karena tadi kita di awal udah bahas, dia berhubungan dengan serbuk sari ya. Jadi dia bisa ditemukan pada sisa-sisa tanaman maupun juga pada sisa-sisa hewan seperti itu, yang bisa jadi itu sebagai polinator gitu. Dan karena polen ini ukurannya sangat kecil, jadi dia sangat ideal untuk dilihat di bawah mikroskop. Kenapa? Dan ini dijadikan sebagai dasar untuk dasar untuk identifikasi gitu. Karena untuk polen ini, karena dia itu beragam bentuknya ya, itu dia bisa ditentukan apakah dia tingkatan taksonnya itu genus, spesies, atau famili seperti itu. Nah, di sini kita bisa lihat polen itu bersumber dari antera. Kemudian kalau misalnya di dalam perbungaan itu kan nanti polen itu akan menjadi penyerbuk ya, untuk polinasi, untuk dia akan menempel di kepala putik. Kalau misalnya terjadi penyerbukan, nanti dia akan menjadi biji, kemudian akan menjadi tanaman gitu. Jadi itu tentang polen itu. Okay,
thank you. So pollen develops basically as a unicellular microspore of seed plants. And here is basically, the then produced multicellular pollen grain is the male gametophytic generation of seed plants as a source of transport, like a source of male gametophytes and transportation units. So basically, pollen, as I mentioned, is produced in the anthers. It starts off as a microsporocyte and then undergoes meiosis and produces this tetrate, which you sometimes even still find in your slides, which then releases the microspore as a unicellular grain, undergoing mitosis and then producing these male gametophytes, gametes, which then basically fertilize the pistils, so the female parts of the flower, through this pollen tube. So you can see also the pictures over here, basically the pollen tube, which releases the male gametes out of the pollen grain, which here you can see is covered by this protective wall, which is so resistant to decay. Okay, you're mute.
Oke, sorry. Jadi perkembangan polen itu dimulai dari, kalau kita lihat kalau di sini kan polen itu berperan sebagai generasi gametosit yang jantan ya. Jadi kalau kita lihat perkembangannya itu mulai dari sebagai mikrosporosit ya pada anther, kemudian berkembang mengalami pembelahan meiosis, kemudian dia berubah menjadi tetrat. Kemudian di tetrat inilah salah satunya itu disebut dengan mikrospora, dan ini akan menjadi sebagai sari yang akan nanti mengalami germinasi ya. Dan kemudian di sini kita bisa lihat di gambar yang ada akar-akarnya ini adalah polen yang sudah memiliki kalau istilah tadi dia bilang itu adalah berkecambah. Jadi kalau pada gambar yang di mikroskop itu ada dua, satu kalau yang bagian bawah ini ya, bagian 10 gambar yang sebelah kiri itu ada satu yang tidak terlihat dinding polennya, itu karena dia sudah berkecambah, sedangkan yang bagian atasnya itu masih terlihat dinding polen.
Yeah, cheers. So when I talk actually here about pollen, pollen can come basically from many different sources when we study it. So we, on the one hand, have fossil pollen, so pollen that comes, for example, from sediment cores, as you can see it over here. So pollen that has been preserved over a long period of time. This is majorly the material that we use, for example, for paleoecology. And that can give us an indication about past vegetation. And then on the other hand, we also have the modern pollen, as maybe you remember our working package 1 and 2, our working package 2 and 3 discusses majorly. So this can be pollen from pollinator organisms, here for example the honeybee, or from pollen traps, as you can see it right next to it, or even just from surfaces. So for example here just a shoe, which is sampled for pollen here.
Jadi untuk polen palynologi itu mempelajari ada dua ya. Ada yang pertama itu fosil polen. Ini adalah polen dari masa lalu. Biasanya kita ngambil dari menggali tanah, dan itu fungsinya adalah untuk melihat ekosistem masa lalu itu seperti apa. Ya, sedangkan untuk modern polen itu kita bisa temukan di yang saat sekarang ini ya. Kalau yang fosil itu di jangka panjang sebelumnya itu. Modern polen itu kita bisa temukan di polinator sendiri seperti lebah, atau kita bikin yang kayak bambu ini adalah perangkap polen, atau kita bisa menemukannya di permukaan atau surface. Itu contohnya itu misalnya di telapak sepatu gitu.
Yeah, thanks. So when we want to observe this pollen, this basically started off in 1830s, not with a pollinologist or a botanist actually, but with an opticist, which was Joseph Jackson Lister. And he did not especially study pollen, but he actually figured out a problem that has bugged people who used microscopes for many, many years before. He basically refined a lens that reduced a persistent blur of color around objects, which was really annoying for people. And after he produced this lens, microscopy made a big step and actually allowed us to observe particles which were far smaller than the particles before. And right away, botanists, including Carl Julius Fritzsche, jumped up on the train. And he was a German person, actually, and took advantage of this technological improvement to advance his studies on pollen, which he noticed before already, but now could study morphologically. And he produced this first work über den Pollen, about pollen, basically. That's this book that you can see on the left hand, only 7 years later, with these very detailed drawings of pollen, which are actually quite accurate already for nowadays. So before, palynology was already a topic. People already knew pollen and they already studied pollen, for example, in medicine, but also natural science. But only after this breakthrough in optics, people were able to study morphological traits of pollen.
Jadi untuk pengamatan polen itu bermula dari penemuan oleh lensa, ya, itu oleh Joseph Jackson pada tahun 1830, di mana dulu mikroskop udah ada, cuman kalau dilihat itu blur gitu. Jadi Joseph Jackson ini dia menemukan cara untuk menghilangkan blur di mikroskop sehingga dapat melihat objek itu secara jelas, dan ini sangat berperan penting sekali dalam perkembangan palynologi. Kemudian ini juga kemajuan dari teknologi ini itu sangat membantu perkembangan morfologi studi morfologi polen seperti itu. Kalau dulu polen itu nggak kelihatan ujung-ujungnya, ya, orang cuma mengira ada duri-duri gitu, tapi ternyata setelah dilihat dengan mikroskop itu ternyata lebih spesifik morfologinya. Dan ini sebenarnya dari dulu itu orang-orang juga sudah mempelajari polen, itu terutama untuk obat-obatan dan juga untuk ilmu pengetahuan alam, ya. Tapi sekarang dengan adanya perkembangan teknologi itu sudah lebih banyak lagi pembahasan tentang morfologi polen.
Okay, thank you. So when we nowadays actually want to observe pollen, we on the one hand can use a compound microscope, so just a light microscope that you might actually know yourself, or a scanning electron microscope, which of course gives us a lot of detail of the actually outer wall surfaces. However, actually when we want to study pollen in a quantitative way, the scanning electron microscope might not be so suitable. So benefits of the scanning electron microscope are, of course, that we can identify pollen grains to a very high degree. So we can actually find out even the species. And also, on the other hand, we don't have to treat the pollen with strong chemicals. So actually, the pollen can be just observed as it is. However, if we really want to reconstruct, for example, past environments, we need to also count a certain amount of pollen so that we have somehow a percentual weight of pollen in one sample. And this cannot be done with the scanning electron microscope. Also, on the other hand, the scanning electron microscope is quite expensive and is a long operating time. So I think like we don't even have it. So yeah, maybe this is not always the option. However, pollen can also easily be studied with a light microscope. And here, actually, the Erdmann acetylysis method has been developed. So Erdmann acetylysis was basically a method to increase the efficiency of pollen analysis, for example, from sediment, but here now again also from, for example, modern pollen traps or pollinator organisms. And these pollen grains here are basically artificially fossilized. So we remove all the organic substances on the surface of the pollen except the sporopollenin, and colorize colorize the pollen wall to maximize their features. So we are able to identify certain morphological traits. And this now is suitable for quantitative analysis. However, our degree of identification is reduced compared to the scanning electron microscopy. So rarely we come to the species level, mostly to the family or genus level. However, it still gives good results. So down here, you actually see the same pollen grain on the one hand with the scanning electron microscope, and on the other hand with the compound microscope.
Jadi untuk observasi polen itu bisa menggunakan mikroskop elektron atau mikroskop kompon atau mikroskop cahaya biasa ya. Kalau untuk mikroskop elektron itu kelebihannya kita bisa melihat permukaannya itu lebih spesifik seperti itu. Jadi dibandingkan mikroskop cahaya itu lebih nampak jelas sehingga kita bisa melihat polen itu, menentukan polen itu sampai spesies mana seperti itu. Dan kemudian juga untuk mikroskop elektron ini juga tidak perlu menggunakan namanya asitolisis ya. Jadi kalau misalnya polennya masih baru itu tidak akan menghancurkan akan dapat merusak polennya. Tapi kalau dia fosil polen itu nggak perlu zat kimia lah intinya di sini ya. Tapi sayangnya untuk mikroskop elektron ini mahal harganya dan butuh beberapa komponen yang harganya lumayan, sehingga dia kurang bagus untuk analisis kuantitatif. Nah, tapi kalau untuk melihat sampai tingkatan spesies itu bagus ya. Nah, jadi untuk analisis polen dengan menggunakan mikroskop elektron itu menggunakan metode asetolisis ya, di mana dikasih larutan kimia namanya aseton yang nanti dia akan membuang semua materi organik yang ada di fosil polen. Nah, sehingga nanti pas dibuang itu dia akan memaksimalkan pencahayaan pada dia di bawah mikroskop. Dan ini bagus sekali untuk digunakan sebagai analisis kuantitatif seperti itu. Nah, kalau kita lihat di dua gambar di bawah, hasilnya akan sangat berbeda antara polen yang dari mikroskop elektron sama mikroskop cahaya. Kalau mikroskop elektron itu sangat spesifik, tapi kalau mikroskop cahaya itu seperti ini saja. Sehingga untuk mikroskop cahaya ini biasanya paling identifikasinya sampai genus atau famili saja. Okay.
Yeah, thank you. So now a bit in detail about the Asman acetylysis. So actually this is a treatment of pollen which uses a lot of chemicals and also is quite aggressive, I would say. So you basically start off already with hydrochloric acid to basically remove all extra material that you find in your sample and to dissolve a lycopodium marker. Then you will treat it with calcium hydroxide, which is also basically to remove organic adhesion and release, for example, sediment a little. Then we have a treatment with hydrofluoric acid. For example, if we have a lot of silica in our samples, which can happen when you take samples from soil, which I think you might know is like one of the strongest acids we find in general. However, it actually doesn't break down sporopollenin. So our pollen wall still remains intact, even though we use all of these very strong acids. Then we use acetic acid and also a solution for acetylysis specifically, which is a combination of sulfuric acid and acetic anhydride. So this also, again, very strong acids together. Yeah, but our pollen prevails, and going through all of these treatments, you can see here on the right hand, still the pollen is intact. You can see the single grains and you can even see the structures of the wall.
Jadi kalau untuk mengamati polen kita pakai HCl 10% ya, itu gunanya untuk menghilangkan material organik. Kemudian juga kita untuk meremove lycopodium, nanti sebenarnya kita akan menambahkan juga lycopodium itu untuk marker. Kemudian kita tambahkan KOH ya, KOH, dan nanti kita akan cuci juga dengan basa asam kuat ya, itu HF, itu asam kuat. Jadi dia itu bisa menghancurkan semua material organik. Tapi kalau untuk polen, dinding polen itu cukup kuat terhadap HF ini gitu. Kemudian kita akan saring juga dengan saringan ukuran 120, kemudian kita kasih asetolisis untuk menghilangkan HF-nya ya. Kemudian baru kita amati. Nah, di gambar sebelah kanan kita bisa lihat itu ada polen, itu ada yang tunggal ya, ada yang bersatu beberapa ya, atau majemuk, tapi kita juga bisa melihat struktur dari dinding polennya.
Okay, thank you. So yeah, I think what comes out of acetylysis is not only, of course, the singular pollen grains and the wall structures to be recognized, but also a wide palette of morphology. So this is actually, I think, what attracted me also the most to this discipline. Basically, the beauty of pollen. So when you look at this, for example, here croton, you have beautiful shapes and colors and wall structures, which I think is also very fascinating about pollen itself, that it can be so diverse and also actually allow us identification. Yeah.
Ya, jadi yang kalau polen itu bentuknya beragam baik dari warna maupun morfologinya. Jadi kecantikan polen-polen inilah yang membuat saya tertarik kenapa mempelajari palynologi ini. Jadi kita karena dia beragam makanya dia bisa jadikan dasar analisis identifikasi. Oke.
So about pollen morphology itself, certainly with this wide range of morphology also comes a wide range of size. So we find pollen actually, which can be large, of bigger than 100 micron, which you can almost see already with the visible eye. And pollen which can be teeny tiny, very small, so look smaller than 10 micron even. Here, for example, the smallest pollen is myosotis. And then, of course, many sizes in between. So this allows you, basically everything, to see everything under the microscope.
Ya, jadi polen itu ukurannya beragam ya. Ada yang ukurannya besar, lebih dari 100 mikron, atau ada juga yang sangat kecil. Kalau yang sangat kecil itu ada yang kecil dari 10 mikron, seperti itu. Nah, kita bisa lihat ini ada myositis, ini ukurannya kecil sekali, tapi ukuran diantaranya juga beragam. Jadi morfologinya juga beragam. Oke.
And pollen doesn't always come alone. So when you actually study pollen grains, you don't always just see the single pollen grains as you saw it before now most of the time. But pollen can also be dispersed in units. So it can be units of two pollen still sticking together, or a very typical pollen unit is also 4 pollen grains, which we then talk about the tetrade, which is actually one of the, if you remember, states after meiosis, and even foliates. So more than 4 pollen grains even sticking together and forming these also quite pretty shapes, I think.
Jadi polen itu nggak selalu jomblo ya, jadi ada juga yang beragam, ada yang menempel berdua, 3, atau malah yang banyak tapi bentuknya nggak teratur, seperti yang foliat yang gambar terakhir. Okay.
And certainly, like our identification, it then depends on actually how this pollen grain is shaped in symmetry. So pollen, if you remember, originally is attached mostly in tetrads, and these tetrads also determine actually different polarity and different symmetry in pollen. So pollen is not just round and all the same from all sides, but it has like different features depending on its polarity. So it's attached in the middle. This is our pole, basically, like you know it from the globe, the Antarctic and the Arctic. So and on the sides, basically have the equator, on which mostly our openings, our perches are located.
Jadi untuk polen itu ini kan dijadikan dasar identifikasi. Jadi salah satu yang paling penting kita ketahui itu adalah simetrinya ya. Jadi polen itu nggak semuanya bulat, tapi ada yang beragam bentuknya, ada yang asimetris juga seperti itu. Dan di sini kita perlu mengetahui kutub-kutub polen itu di mana ya. Di sini di gambar yang B itu dilihatkan itu ada bagian ekuator, kita harus tahu bagian kutub utaranya yang mana, bagian kutub selatannya yang mana. Tapi kita menyebutnya nggak kutub utara, kutub selatan ya, tapi kita menyebutnya kutub aja seperti itu. Oke.
So here a bit about the polarity, about the symmetry. So again, you can see here the poles. So basically alongside through the poles is our polar axis. And then we have the equator in which we mostly find our perches. This is this brown shapes here. It's supposed to be the openings of the pollen grain. I come to a perches later still, because it's also an important thing, I think, to know about pollen, that they have this. And this polarity, the symmetry can cause different shapes in pollen. Of course, pollen is not always round and circular, but it can have many different shapes. It can be isopolar, so actually quite regular, but it can also be heteropolar. So the lower part, for example, of the pollen grain can be bigger than the upper part. Makes it tricky to identify sometimes, but I think maybe also good to know.
Yes, very tricky. Jadi di sini saya bilang bahwa penting sekali untuk mengetahui polarity itu kutubnya yang di mana ya, sama simetri dari polen itu. Karena polen itu tidak selalu bulat, jadi kita harus untuk mengidentifikasinya itu kita mesti mengenal itu polaritasnya seperti apa. Karena untuk semua polen itu beragam ya. Jadi misalnya kita harus tahu dulu bagian ekuatornya yang mana, kutubnya yang mana gitu. Dan dari sana kita bisa menentukan, oh ini dia isopolar atau heteropolar.
Mm-hmm. And then again, not only along the polar axis, there can be differences in shape. So also, of course, it depends on the ratio between the polar axis and the equatorial axis. So the equatorial axis can also be wider. And here we differentiate mostly between oblate pollen, which then is the polar axis that is smaller than the equatorial axis, spheroidal pollen, where it's the same, so it's basically completely circular, and prolate, where the polar axis is bigger than the equatorial axis.
Jadi untuk melihat bentuk polen, di sini kita perlu melihat perbandingan antara polarnya ya, aksis itu garis antara polarnya sama diameternya, diameter ekuatornya. Jadi itu akan menentukan tipikal polennya yang mana. Kalau misalnya yang pertama itu yang oblate ya, itu kita ketahui bahwa polarnya itu lebih kecil dibandingkan ekuatornya. Sedangkan kalau dia spheroid itu ukuran polarnya ya itu sama gitu. Sedangkan untuk yang prolate itu ya polarnya itu lebih besar dibandingkan diameter ekuator. Jadi kita perlu mengetahui perbandingannya seperti itu.
Mm-hmm. But it's not only that, of course, there's many states in between. So again, pollen is so diverse morphologically that there's shapes in all different kind of sizes. So this can be from many different stages in between, over suboblates, preoblates, subprolate, and so on. So yeah, I think as the university has experienced how diverse the shapes of pollen grains can be.
Yes. Jadi polen itu sangat diversity-nya itu, keragamannya itu sangat banyak ya. Jadi tidak hanya 3 tipe tadi, tapi ada juga tipe subnya gitu. Misalnya di sini ada oblet, suboblet, gitu, semuanya beragam gitu. Jadi sebenarnya bilang karena saya udah pernah ngelihat polen sebelumnya itu memang benar-benar sangat beragam. Okay.
Yes. So yeah, let's get back maybe to the apertures. So basically, like here, for example, I just took some examples of apertures scanned with the electron microscope and colored a little. Here actually in function, so you can see apertures can be an exiting point, for example, for the pollen tube.
Okay. Ya, jadi ada bukaan polen namanya pollen aperture. Itu kita dapat temukan, kalau yang saya lihat ini itu dilihatnya dengan mikroskop elektron. Bukaan polen ini atau pollen aperture ini adalah tempat di mana buluh serbuk itu nanti keluar seperti itu.
But this is not the only function. So in general, we define the aperture as the region of the pollen wall which is morphologically different or anatomically different from the rest of the pollen wall. So it generally is presumed, so we think, to function as a germination site for example, here for the pollen tube. However, it can also have other functions, for example, the egress and ingress of water. during desiccation and hydration. So while the pollen is transported, it can actually shrink and or take up water to basically deal with different environmental conditions. Another point is that it's maybe also a structural component, so that actually the pollen wall doesn't break during such stress that comes with water intake or uptake. For example, this grain shrinkage. And then, of course, the major function probably is the opening to exit the pollen tube.
Ya, jadi aperture atau bukaan ini itu juga sangat beragam ya, tidak hanya bentuk tapi bukaannya juga. Jadi aperture ini memiliki beberapa fungsi yang utama. Yang pertama itu adalah untuk memasukkan dan mengeluarkan air dari polen itu sehingga dia bisa bertahan dengan kondisi lingkungannya. Yang kedua, aperture ini juga berfungsi sebagai komponen yang bisa mendistribusikan tegangan. Jadi kalau misalnya ada kondisi stres lingkungan yang misalnya itu penyusutan dari polen, jadi dia juga bisa menyesuaikan seperti itu sehingga dia untuk beradaptasi dengan lingkungannya. Kemudian fungsi utamanya itu adalah tentu untuk tempat keluarnya bulu serbuk.
So these apertures can be very diverse, as maybe here already seen on the right side. So these openings can have different shapes, and this is also actually developed differently by every plant group. So we find a different type of shape, a different form in many different groups, and this helps us actually to identify different groups. So some are parallelly developed, so it's not always correlated to one specific group. So one group can have also different type of apertures, but in general, this helps us to differentiate here. So three major aperture types are actually the porous, so a circular opening. The form is completely round, and mostly the position is equatorial. As I mentioned before, there's always like this orientation in polarity where we can find most of the apertures, or it can also be globally. For example, here in Arenaria, we can find it all over the pollen grain. Then we have the corpus, which is like a slit. It's like an elongated opening. This can also be equatorial, but also global. And one more is the corpus. So actually, this is a combination of a pore and a corpus. So we have the circular opening in the middle, mostly on the equator. And then we have, again, this elongated opening, as we saw it before, mostly also oriented on the equator.
Jadi untuk bukaan polen ini kan beragam juga ya, yang ada tiga tipe di sini. Yang pertama itu ada porus ya, bentuknya itu membulat dan posisinya itu ada di bidang ekuator. Jadi bisa jadi satu aja dia, atau dia bisa berada di banyak titik atau global seperti itu. Ada juga yang bentuknya kolpus atau memanjang, kita bisa lihat di gambar Luciata itu, dan posisinya juga macam-macam ya. Ada yang di ekuator saja, atau ada yang dia menyebar seperti itu. Atau yang berikutnya ada kolporus. Kolporus itu dia gabungan antara porus dan kolpus ya. Jadi kemudian dia bisa juga membentuk semacam gelembungan seperti itu, dan posisinya sama. Ada yang di tanaman tertentu itu posisinya hanya di bidang ekuator, dan di tanaman polen lain itu dia bersifat global.
Yes. And then, of course, there's different amounts of apertures. So not all apertures are three or all the same, but they can come in different amounts. I think the most common is 3. Then you would add a 3 in the description of your pollen grain. So when you describe the pollen grain, you also need to mention which type of aperture it has and how many. And we abbreviate this with, for example, here 3 corporate. But it can also be 2, so D, 4, so tetra, 5, penta, and so on. Right? So yeah, it always depends on your type of pollen that you have in front of you, but it can help you to actually come to, for example, a genus, a certain type of genus.
Jadi untuk polen sendiri itu aperture itu sangat penting ya untuk identifikasi tadi. Jadi kita bisa mengidentifikasi bukaan polen kita itu dengan melihat jumlahnya berapa. Kebanyakan itu jumlahnya 3, tapi tidak semuanya 3. Kita bisa akan menamai polen kita itu berdasarkan aperture-nya dengan menambahkan di depannya itu di ya atau di untuk 2 bukaan, tri untuk 3 bukaan, tetra untuk 4, penta untuk 5, heksa untuk 6 dan seterusnya seperti itu. Jadi tergantung banyak bukaan polen. Okay.
Yeah, and then of course we have again our morphological diversity. So you can see here just some examples for aperture types in pollen, and you can see actually this list still goes on. So this is not all. We can have just the pores here, or a different combination of pore and colpi, which can sometimes look very funky, as you can see it on here, the festate. They can look very pretty or even circulate around the entire grain. So yeah, wide morphological differences, but it helps us to identify pollen grains quite well. Yes.
Jadi morfologi polen ini sangat beragam ya. Jadi ini baru sebagian dari beberapa morfologi berdasarkan bukaan atau aperture-nya gitu. Jadi di sini ada yang bentuknya, dia bilang tadi ada yang imut ya, ada yang lucu, ada yang funky seperti itu. Dan ini ada yang bagus, cantik seperti itu. Jadi macam-macam dari bentuk polennya berdasarkan bukaannya. Jadi kita bisa merujuk nih posisinya ada bukaan itu ada di mana. Oke.
I just took the liberty to put some examples in here. Again, the comparison between the electron microscope and the compound microscope. So you can see again how acetylysis works that way. So this is again always the same pollen, at least the same pollen type, just from different scanning methods. Yeah, and you can see again here, for example, just a pollen grain with just one single pore, which here comes from Poaceae, or just 3 pores here from Betula pendula, or actually pores distributed globally throughout the entire pollen grain here from Chenopodium viridum, which you know already from my demonstration of the acetylysis method before.
Ya, jadi ini perbandingan antara polen yang di atas itu dengan mikroskop elektron, di bawah itu dengan mikroskop cahaya saja dengan pembagian asetolisis tadi. Jadi untuk ya kita bisa melihat nih ini adalah spesies yang sama, sampel yang sama seperti itu. Yang pertama itu dari Poaceae itu dia memiliki satu lubang aja apa sih namanya tadi. Terus yang kedua itu ada yang dia memiliki 3 bukaan ya, 3 pori gitu tadi dibilang, 3 pori. Misalnya itu betula ini, kemudian ada yang dia tersebar secara global atau menyeluruh gitu. Jadi yang di atas itu dari mikroskop elektronnya terlihat, yang di bawah juga terlihat dengan mikroskop cahaya, cuman bentuknya berbeda. Oke.
Yeah, thank you. I just give some examples still now where it's not so regular. So what you can actually face when you see pollen, that not all apertures are neat and fit to our schemata. But this, for example, here is monocycate pollen. This is pollen that you find from monocotyledons. This is actually the most basic pollen, I think. So this is like one single opening, one single here called sulcus, that opens just alongside the entire pollen grain, along the polar axis. Ya.
Ya, jadi di sini adalah bentuk arah kita melihat polen itu berbeda-beda ya. Jadi ini sebenarnya adalah satu polen. Ini adalah polen dari monokotil ya, namanya monosukat polen ya. Nah, jadi kita kalau misalnya lihat dari kutubnya bentuknya seperti gambar yang pertama. Tapi kalau kita lihat dari posisi distal itu beda lagi bentuknya. Atau di bidang ekuator kita lihat itu bentuk gambar yang ketiga. ketiga dan gambar yang terakhir itu kalau misalnya kita lihat di sisi ekuator yang lainnya. Jadi untuk satu tanaman itu bidang kita pandangnya itu akan memberikan bentuk yang berbeda. Jadi untuk mengamati polen nanti kita harus membayangkan ini bagian mana. Oke.
Yes. So like this is just some examples of different monocycate pollen. Actually, I just wanted to show that this is like all the same aperture type, but still like the pollen grain can look quite different.
Ya, jadi beda ya kalau yang tadi itu satu monosoket, monocotyledon. Kalau ini dari ya, tanaman yang berbeda seperti ini. Iya, tapi bentuknya sama. Oke.
Then another maybe strange pollen grain that you might encounter if you work with pollen is actually saccade pollen. So this is actually a pollen grain here in the middle. So what is like a little brighter here again with a scanning electron microscope. And it has two basically air sacs on the side for floating. So this is actually a pollen grain which can travel very far distances through several kilometers, like several hundred kilometers even, with the wind. And this comes from gymnosperms. So for example, maybe you know Podocarpus, or here Dacrydium, which I think also occurs on Sumatra.
Yeah. Jadi ini adalah bentuk polen yang agak berbeda ya daripada yang sebelum-sebelumnya, itu nggak masuk kriteria. Jadi dia punya bagian pinggir itu yang memungkinkan dia itu dibawa angin jauh gitu. Bahkan tadi dibilang sampai ratusan kilometer seperti itu. Jadi dan ini beberapanya ini ada tanaman yang ditemukan di Sumatera.
Yes. And then besides pollen, there's also spores. So we don't only study pollen grains, but also the grains from pteridophytes. So for example, mosses and fern. And here you can see there's basically two major types of spores. One is the trilete spore, which has here called a lesura, which is three that are combined in the middle. So it always forms this triangle. And on the other hand, the monolith spore, which just has one lesura, which opens alongside the entire pole, similar to the monocotyledon pollen that we saw before.
Ya, jadi ini adalah spora dari Heterodepita ya, lumut dan juga paku-pakuan. Jadi ada dua spora utama. Di sini ada yang trilet dan monolet. Nah, kalau untuk Heterodepita kita melihat lesornya di sini atau apa ya, belahannya seperti itu ya. Jadi ada yang tiga, ada yang cuma satu. Jadi kalau yang satu disebut dengan monolet, kalau yang tiga disebut dengan trilet spores.
Yes. Which leads me now to actually like one of our last morphological features, which is the pollen wall itself. So pollen wall is composed majorly, as I mentioned before, out of sporopollenin, which is this organic compound which is so resistant to decay, to acids, to weathering, to everything. It encloses the cytoplasm. So inside of the pollen grain, usually you have the cytoplasm, as you can see it in the lower picture here on the right. And when we now study the pollen grains, usually the cytoplasm inside is already removed with time, because sometimes we study fossil pollen, there everything has already decayed except the sporopollenin. But when we also study modern pollen, we usually give it a chemical treatment, which also removes the cytoplasm from the inside. And this pollen grain wall offers certainly protection against, for example, UV radiation, but also drying and mechanical damage, for this to be the perfect dispersal unit. It is laminated, so there is different layers of inside of the pollen wall, which I come to later. And it's often quite fiercely ornamented. So you can see here quite the flashy example with this echinate pollen wall, these little spikes that come from it as an ornamentation.
Ya, jadi yang berikutnya kita lihat itu dinding dari polen atau serbuk sari ini itu bentuknya sangat beragam ya. Jadi dia itu fungsinya adalah untuk pelindung, dan di bawah dari dinding serbuk sari atau dinding polen ini itu ada sitoplasma sama dengan sel biasanya gitu. Sehingga dia fungsinya sebagai pelindung sitoplasma. Selain itu fungsinya itu adalah untuk melindungi dari radiasi UV atau kekeringan ya, atau kerusakan mekanis, misalnya ada pukulan atau jatuh segala macam. Kemudian dia juga fungsinya sebagai laminating, atau dia sebenarnya di bawah dari pollen wall itu dia memiliki lapisan-lapisan yang berbeda ya. Tapi biasanya ini akan menghilang kalau udah dikasih zat kimia. Dan juga ada beberapa ornamen pada dinding polen juga, terutama untuk polen-polen yang modern itu bisa juga tidak kelihatan sih pada saat pemberian zat kimia. Okay.
Okay. So this pollen wall or the pollen surface, let's say, can in general be divided into 3 different regions within the relative amount of each layer, varying between the species. So every layer basically can be unique for a different genus or for a different species, which then again allows us identification. So we have this outer exene wall, which is often multi-layered and is mostly composed out of this chemically resistant polymer, which is the sporopollenin, and which also contains our apertures and all these different morphological features. And then we have the inner intrine, which usually is also multi-layered, but majorly made out of cellulose, which we can still find also after decay. However, sometimes it is removed already with time or with chemicals. And then we have the pollen coat, which is mostly a coat composed of lipids and proteins, but also sometimes pigments. However, this pollen coat we remove, for example, with acetylysis. So this can still be seen with electron microscope, but when we give it chemical treatment, we try to remove it to open up these morphological features of the exene, so that we can see all the structures. But the pollen coat, of course, is there when you watch live pollen.
Iya. Jadi untuk dinding polen itu ada 3 bagian ya. Ada yang pertama itu outer exene-nya, yaitu dia biasanya terdiri atas lapisan multilayer juga, dan di sini terdapat aperture tadi yang sudah kita pelajari. Kemudian yang berikutnya itu ada yang namanya inner intrine, itu merupakan lapisan multilayer juga, tapi dia selulosa. Dan yang terakhir itu ada namanya pollen coat ya, atau pembungkus polen. Ini terdiri atas lipid, protein dan segala macam, tapi pas kita pewarnaan untuk melihat di bawah mikroskop kompon atau mikroskop cahaya, itu dia akan menghilang. Tapi kalau dengan menggunakan mikroskop elektron itu dia masih ada, masih terlihat.
Thank you. So this is like a little bit of a scheme of the upper 2 layers. So we have the exene, which you can see here. It's multi-layered. Often we have different layers, which can again give us indications about the morphology, and then again about the identification. Not every pollen grain contains all of these layers. So here, for example, you can see one where you have a tectum. So this is like the upper part, which often looks very, very interesting and very fancy when you look at it under the microscope. But then again, we can have some pollen grains which don't have this tectum, for example, but which have wall features which come from the here columnella. So this is like one of the layers of the exene. And they can then again cause the pollen wall and the surface to look very different. So here, for example, you have this echinae maybe, and here you have this like little columnae that look out of the pollen grain. So this is important to look at. It's important to try to find these layers and to identify and measure them. And then, of course, we have the inner wall, which is the intine. And this is interesting also in terms of how the exine and the intine move together. So how the exine is bigger, maybe, or smaller than the intine can give us indications again about identification. So this causes a lot of diversity in pollen.
Oke, jadi di dinding polen ini dia itu beragam ya. Tadi ada yang paling luar itu eksin, jadi dia berlapis. Nah, untuk dinding polen yang eksin ini pun dia memiliki karakteristik masing-masing gitu. Jadi ada yang punya benjolan seperti ini ya, ada yang bentuknya seperti seksin seperti yang bulat tadi yang ditunjuk, kemudian ada intin yang bagian bawah. Jadi intin dan eksin itu bisa ketebalannya berbeda-beda. Jadi dengan ketebalan yang berbeda-beda ini menjadi dasar untuk mengidentifikasi polen. Svea, may I ask before ini, because before I forget. So when we want to see the pollen wall, we have to see by microscope electron.
I'm sorry, again?
If we want to see or identify the pollen wall, by to know how thick the exine or intine, so it means we have to see under the electron microscope.
Under the electron microscope, it's most detailed and most clear, but it's also possible to see it with the compound microscope. So with most pollen grains, I can see at least the intine and the exine. Yeah.
So for the ratio between exine and intine, could we see in under a compound microscope?
Yeah, you can see it there. It's a huge help for identification actually. Yes.
Jadi di sini saya bilang, jadi untuk melihat exine dan intine ini tidak harus pakai mikroskop elektron. Jadi dengan mikroskop cahaya pun dia bisa melihat kira-kira rasio antara exine dan intine itu berapa. Oke.
Yes, thank you. Just for a bit of detail. Yeah, here maybe still in 3D, so you can like imagine this a little better when you actually look at the surface, but it's to demonstrate. So now when we actually go into, like we have identified all of these morphological features, right? We have seen all the different parts that are important to us, and maybe we made some notes. Now we can actually go into pollen identification. So we can try to find out what we actually see, like what is the pollen grain. And for that, it's sometimes helpful to use such online databases. So there's by now already quite a lot of databases. It's still sparse because not so many people work with pollen, but some of them are very detailed, like just a bit of an advice here is to use the Australian Pollen and Spore Atlas, which also covers pollen from Indonesia. And here you can actually put in, for example, how long your equatorial axis is, what surface pattern you identified. You can maybe still say the morphology, which here means the amount of apertures. You can even specify the floristic region, which would be Southeast Asia here. Or you can simply search for a pollen type from one genus that you can put here into the search term.
Ya, jadi untuk identifikasi polen itu kita bisa merujuk dari beberapa website ya. Jadi ada yang Global Pollen Project, ada yang dari Australia, atau kita bisa juga memasukkan data informasi polen kita ke sana. Misalnya kita punya polen dengan aperture-nya jumlahnya berapa, kemudian posisi dari bukaannya itu ada di mana. Jadi kita bisa memasukkan informasi-informasi polen yang kita punya juga ke database ini. Oke?
But of course, there's also literature. So, for example, just to mention here, the pollen flora of Taiwan, which I think also covers most of the pollen that you also find, for example, on Sumatra, with a nice picture band where you can see also a lot of pictures of pollen. But this also goes for peridophyte spores, of course. So we also have books which discuss the different morphological traits of spores. So it's always interesting to look into the books also, because sometimes it's nice to have something in hand and to actually just browse through pictures. And Of course, also use papers that you find. There's a lot of papers which only discuss morphology. So you have a lot of pictures of different pollen from specific botanical regions, actually, which can help you identify your pollen.
Jadi untuk identifikasi polen ini kita juga bisa menggunakan berbagai macam buku. Misalnya ini ada buku Pollen Flora of Taiwan. Nanti kita akan membandingkan polen yang kita dapat di bawah mikroskop sama gambar-gambar polen yang di sini. Atau misalnya spora Pteridophyta untuk tanaman-tanaman paku-pakuan atau Pteridophyta. Atau kita juga bisa mendapatkan dari artikel jurnal. Terus gambar-gambarnya yang ada di sana kita bandingkan dengan hasil polen yang kita temukan di bawah mikroskop.
Yes. But enough about identification now. Maybe let's get back to palynology in general and how to use palynology. But I should start off actually with how modern palynology came to pass, because nowadays we work quite differently to what we saw from the 18th century with how they worked with the new lens. Now we have developed already much further, and it actually basically started palynology itself as a discipline in 1916 with the 16th Scandinavian meeting of natural scientists in Oslo. And here Lennart van Post actually presented the first pollen diagram. A pollen diagram you can see down here below. I will still explain what actually this means. So a pollen diagram is like basically a series of graphs for different plant taxa, which are then displayed side by side. So each of the single lines here is a plant taxon, or at least the pollen from it. And the horizontal axis, so the x-axis, shows the relative abundance of pollen, so the percentual amount of pollen that you found in your sample. So let's say one area of this line is one sample. And here we found, for example, 20% of this taxon number 1, and 40% of the taxon number 2, and 60% of the taxon number 3 or something. For each of these taxa, for each of these plant types, you can find this relative abundance, and also the y-axis here then shows the age through time, or the depth of the sediment where we took the sample. So actually alongside the y-axis you find the samples displayed. It can also just simply be a sample number. Okay?
Jadi kita balik lagi ke palynologi ya. Jadi sekarang untuk di palynologi itu kita melihat polen diagram ya. Polen diagram itu tadi yang ditemukan dimulai dari tahun 1916 tadi, itu awalnya dari ilmu palynologi ini menjadi satu bidang ilmu seperti itu. Nah, ini adalah salah satu contoh analisis polen yang tadi ditemukan di awal ya, di mana kita bisa melihat di sumbu horizontal yang bawah sumbu X ya, itu menunjukkan kelimpahan polennya itu seperti apa. Dan kita bisa melihat grafik-grafiknya itu ya, jendul-jendul gitu, itu makin tinggi dia itu berarti makin berlimpah si polen itu. Nah, sedangkan untuk ke atas, jadi kan ada grafiknya itu ada warna hijau, warna pink, warna kuning ya, itu menunjukkan jenis-jenis tanamannya. Jadi kita bisa lihat di sini kalau yang kuning itu tanamannya kelimpahannya lebih sedikit dari yang berwarna hijau yang di akhir-akhir seperti itu. Jadi kita bisa melihat di sana, dan di sini kita juga bisa melihat ya grafik di sumbu Y itu waktunya kita juga bisa meletakkan di sana. Jadi ada yang mereka menambahkan waktunya. Jadi makin ke bawah itu kita lihat untuk grafik tanaman yang di alnos misalnya di sana itu jumlahnya banyak sekali ya. Kemudian makin ke atas itu sedikit, atau ada beberapa masa dia itu menjadi lebih sedikit. Kita bisa melihat di waktunya. Jadi misalnya pada tahun 3000 itu jumlahnya tidak begitu banyak, 3000 tahun yang lalu ya. Tapi pada tahun dekat-dekat sekarang itu malah dia tidak ada lagi seperti itu, atau jumlahnya sedikit. Jadi kita membaca polen ini berdasarkan sumbu Y, sumbu X, dan juga informasi yang bisa ditambahkan. Okay.
Mm-hmm. So yeah, let's get back to Lennart van Post. So we have to imagine it was like 1916, and this was actually the time of the First World War. So the reason why Leonard van Post produced this pollen diagram was actually that Sweden made him study the peat deposits of the country because they did not have a big availability of fuel. Because of the First World War, they had problems with transportation and also problems with basically using fuel for the households and not for the war. And yeah, the country of Sweden basically told Leonard van Post, let's look at our peat deposits. We can use peat for burning because winter was coming. And he was to study how much peat was actually distributed throughout the country. And in peat, of course, we find pollen because it is resistant to decay. So he actually took peat profiles to study the composition of different pollen taxa and therefore the vegetation in the past to see how vast the pollen deposits, the peat deposits, actually were, because he could see how old they were.
Jadi kalau yang tadi ini kan gambar polen di Skandinavia ya beberapa tahun yang lalu gitu. Jadi kalau dulu mungkin dia melihatnya itu agak susah gitu ya karena ada beberapa kondisi mungkin ya. Jadi untuk diagram polen ini mereka mengambilnya di lahan gambut. Jadi melihat dari mereka ingin tahu itu lahan gambut itu pernah terbakar itu tahun berapa gitu. Jadi kita melihat dari kelimpahan polennya. Kalau misalnya kelimpahan polennya itu sedikit, berarti itu mengalami pengurangan seperti itu. Tapi kalau misalnya di zaman itu banyak ya, itu artinya tanaman-tanaman itu berkembang dengan bagus. Dan ini bisa membandingkan kondisi tanah gambut di masa lalu dan masa sekarang.
Yeah, so he presented the pollen diagrams and everyone was really excited. So people were really interested in his studies and they followed him. And this also advanced actually the study in palynology. So many events followed, which were keystones for palynologists. Here, maybe just some to mention, maybe this is a bit out of context for you. But on the one hand, we had advances in chronology. So actually the time frames that we find in our pollen diagrams and how they are classified, which were developed mostly by Blitz Sernander. But then like Rudolf, for example, he came after the Scandinavian meeting of natural scientists and he produced more detailed chronologies, especially also, for example, for trees in Europe, for the different phases of trees after the glaciation. And then we have, for example, the coal industry suddenly becoming very interested, of course, only 4 years later. So the American geologist Jason actually assumed that spores from, for example, Pennsylvanian coals here might as well be used for stratigraphical correlation. So to actually find out where we find coal deposits, which is, I think, still done nowadays, also with pollen. And also, one other example is that the study of honey actually began to flourish again right after this meeting. So here the microscopic examination of honey to majorly look for the quality. And this is developed majorly also in Germany by Armbruster and Onike in 1929.
Jadi diagram polen itu tadi sebenarnya memiliki banyak sekali informasi menarik ya. Jadi informasi menarik itu bisa didapatkan dari banyak informasi dan di sini ada beberapa informasi yang juga mendukung dari polen diagram tadi. Misalnya yang ini ada berkembangnya pada tahun 1920 kan itu lagi berkembangnya industri batu bara ya, dan di sana ternyata ditemukan stratifikasi atau tingkatan-tingkatan tanah itu, dan itu juga memperlihatkan kondisi spora. Jadi kalau kita tahu stratifikasi tanah itu berdasarkan tingkatan tahunnya, maka kita bisa mendeteksi itu spora itu dari tahun berapa. Dan kemudian sekarang juga untuk ilmu tentang polen ini juga ingin mengetahui bagaimana lebah itu dia menyerbuk tanaman apa saja gitu, karena kita bisa melihat polen yang ada di lebah itu atau misalnya di madu ya. Jadi kita tahu itu perkembangannya itu seperti apa, atau kita bisa tahu madu itu mulai berkembang dari tahun berapa. Jadi ini sangat menarik sekali untuk melihat sejarahnya baik ekologi maupun perkembangan madu di sini. Okay.
Thank you. Yeah, and then basically it accelerated every year. There were like new developments. So we had greater insights into the actual pollen grain wall, which you already like learned about. And then soon after, the first book on palynology was published called Pollen Grains, Their Structure, Identification and Significance in Science and Medicine. And also the first pollen analysis circular, so like basically the first pollen paper. This was in 1943, and now actually we are facing the Second World War, so pollen went through some world wars, let's say. And here actually there was the issue that they could not basically communicate well among scientists during this time. So like a palynologist actually was quite upset about that. He wanted to discuss with his colleagues about pollen. So he started to write letters to his colleagues to further discuss issues that come with palynology. And this was then the origin of the circular. So they continued this then every year to basically, discuss like what scientists do, you know, like always talking about new things that they discovered.
Jadi kemunculan palynologi ini itu berhubungan juga dengan saintis-saintis yang ini dari tahun ke tahun ini kan ada aja penemuan-penemuannya gitu. Jadi mereka yang berada di daerah yang berbeda ya, atau dia bilang college, tadi ya teman-teman kampus atau teman-teman yang sama bidangnya itu saling menyurati gitu. Jadi mereka memberikan informasi ini di polen apa yang ditemukan, ini polen apa yang ditemukan. Jadi sejak itu berkembanglah ilmu palynologi itu karena ternyata banyak hal menarik yang ditemukan dari polen-polen ini.
Yeah, thank you. And yeah, of course it goes on, right? So there's new developments every day in palynology. Yeah. And nowadays, actually, we use palynology in quite a lot of different fields. So here I picked out some examples for you to see how palynology is applied, or how for example, it can be interesting maybe for you, for your career, to decide if you want to work with palynology and if you maybe want to work in those fields. So on the one hand, we have the petroleum industries, we have forensics and criminology, medicinal palynology for food security, aerobiology, archaeological palynology, biogeography, paleoecology and climate change, and also plant identification and taxonomy. You want to mention?
Yeah. Jadi di sini banyak sekali ya kegunaan palynologi ini. Jadi ada yang dimanfaatkan untuk industri petroleum, ada yang nanti untuk menyelidiki kasus-kasus forensik dan kriminal, ada yang di aerobiologi, ada yang untuk melisopanilogi, yaitu tentang lebah ya. Kemudian ada juga tentang arkeologi. Nah, ini menarik juga ini. Ada yang sekarang banyak digunakan juga untuk identifikasi dan taksonomi ya. Ada juga yang untuk mengetahui biogeografi, paleoekologi dan perubahan cuaca. Jadi banyak sekali fungsinya dari palynologi ini. Jadi kalau kalian ada yang tertarik kerja di palynologi, ini kalian bisa melihat nih satu bidang yang mau diambil itu yang mana.
Yeah, I will go in more detail about the different disciplines. So you can get a little bit of an insight. Of course, I'm not an expert for all these disciplines. So I mostly come from paleoecology, maybe a little bit of biogeography and archaeological palynology. But I hope that I can give you a little bit of an idea what we are working with. So let's maybe start with the plant identification and taxonomy. So this is actually also where you mostly come from, right? What you're mostly studying. So actually, pollen itself can also be used for morphological characterization. to then indicate taxonomy and phylogeny of different closer related groups of plants. So for example, here, Ophioglossum has been studied palynologically free from the pteridophytes, and a new species has been revealed through the palynological features. This was only, I think, in 2019. However, actually, I have to say here that this has been mostly replaced by DNA metabarcoding. So actually, this is not so common anymore to use palynological traits. In the recent years, you maybe face it yourself. This has accelerated a lot, and we mostly use the DNA to define closer phylogenetic groups.
Jadi yang pertama itu penggunaan palynologi itu adalah untuk identifikasi tanaman dan taksonomi. Jadi dulu palynologi itu digunakan untuk mengkarakterisasi morfologi dari suatu tanaman itu seperti apa gitu. Jadi kita untuk melihat taksonominya seperti apa, filogeninya seperti apa, seperti itu. Jadi kedekatan antara satu tanaman dengan tanaman lain itu seperti apa gitu. Nah, tapi sekarang sudah ada yang namanya DNA metabarcoding, dan ini lebih cocok untuk menentukan tingkat filogeni ya atau kekerabatan dari satu tanaman dibandingkan dengan palynologi. Tapi untuk identifikasi secara morfologi itu masih, tapi lebih gampang dengan menggunakan metabarcoding.
So let's get a bit into archaeological palynology. So palynology can also help archaeologists to uncover maybe some mysteries that they are still facing. And here palynological stratigraphical analysis can be employed to study, for example, past anthropogenic interactions with the environment, including, just an example here, food resources, or like different plants used in rituals, and so on. Just one example maybe here is that they actually found pollen in the teeth of Neanderthals, and the pollen was quite diverse. So despite what they thought before, that Neanderthals mostly ate meat and actually didn't eat much vegetables, they could say that actually the Neanderthals had quite a balanced diet and actually ate a lot of vegetables from many different plants. And another example is these pollen found in a burial site in Shanidar, which indicated different flowers, different flowering plants, which quite ornamental flowers, which also sometimes were like perfumed to use for burial rituals in the past.
Jadi dalam berikutnya itu palynologi ini bisa dilihat dalam arkeologi ya, jadi sejarah masa lalu gitu. Jadi kita lihat gambar pertama ini, kita bisa menentukan ini makanan manusia zaman lalu itu apa gitu. Jadi kita bisa membantah teori yang sudah ada. Misalnya nih dahulu diketahui bahwa Neanderthal itu lebih banyak makan daging dibandingkan makan ini ya, makan tumbuh-tumbuhan, jadi nggak makan sayur gitu. Ternyata pas ditemukan penemuan bahwa ditemukan polen di giginya si Neanderthal ini, diketahui bahwa ternyata di manusia Neanderthal ini memiliki diet yang bagus seperti itu. Jadi dia balance antara makan sayur sama makan yang daging-dagingan juga. Kemudian ditemukannya juga di daerah tempat makhluk hidup itu ada polen beragam, itu bisa diketahui juga bahwa si manusia zaman lalu itu dia memiliki ritual atau memanfaatkan tanaman itu untuk apa. Jadi bisa tahu ini manusia purba ini udah pakai parfum atau menggunakan tanaman ini untuk seperti apa, untuk sumber makanan atau lain. Oke.
Then let's get to biogeography. Here, pollen from Paleozoic sediments can be used to actually reconstruct vegetation changes here on the geological timescale, covering millennia of past vegetation distribution and also climate variation, which then can be reconstructed on a certain site where the paleo sediment has been taken from, for example, a lake or a peat bog. And this can help to discuss modern ecological and also biogeographical theories. For example, let's say how certain tropical plant groups have expanded in the past and actually arrived at certain areas, and for example also island formation, and how they were disconnected also in a botanical way. And these theories have already been tested and are still tested using palynological analysis.
Yang berikutnya, analisis palynologi ini juga untuk bisa mengetahui tentang biogeografi suatu vegetasi. Gitu. Misalnya, jadi kalau ditemukannya polen dalam suatu sedimen itu bisa melihat ini vegetasi itu perubahannya seperti apa. Misalnya ini ada pembentukan pulau gitu. Jadi ada tanaman di daerah utara itu bagaimana menyebar ke daerah selatan, atau sebaliknya bagaimana pulau itu terpisah pada suatu waktu sehingga masih ditemukan tanaman yang sama atau tanaman yang sejenis antara satu sisi dengan satu sisi yang lain. Okay, Sophia, it's already 5:30. It is okay to continue more than 1 hour.
For me, it's okay. I don't know. How about you? Like,
okay.
I mean, for me, it's still
okay
early in the day.
About 30 minutes again. Yeah.
Yeah. Okay.
Yeah.
Yes. Okay, so let's get to paleoecology and climate change. So here, majorly, palynological analysis can be used to reconstruct long-term vegetation, but also climate dynamics in the past thousands of years. And pollen here is used as an indicator.
Jadi untuk paleoekologi juga bisa melihat perubahan cuaca pada masa lalu seperti itu ya. Jadi kalau ada kondisi polen yang berubah itu bisa kita tahu nih, oh pada masa lalu itu pernah terjadi kekeringan, pernah terjadi kebakaran atau perubahan-perubahan kondisi lingkungan.
And one example here, for example, is to use pollen in prediction models, which are basically used, for example, for general climate trends, and they can be modified with past climatic reconstructions derived from palynological data, which, for example, already increased the accuracy of future predictions for glacial maxima.
Ya, jadi data palynologi ini juga bisa digunakan untuk memprediksi kondisi cuaca masa lalu. Kita bisa lihat di grafik ini, dia itu menggunakan data-data polen ya, di masa lalu.
Then maybe one more industrial area of the use of pollen is the oil and gas exploration. So actually we can use pollen, but also palynomorphs, for example, dinoflagellates, to identify past petrostratigraphical and geological layers in which we found different ecosystems that then might be the origin of fossil fuel sources. So gas deposits or oil deposits can be found like this, and palynology is here used to map these environments through time and also through space. Here just maybe shortly explained, because I'm also not an expert for this, but in general we use, for example, pollen that can come from the land, so terrestrial pollen, to identify different areas of forests or other ecosystems on the land. And we use dinoflagellates for marine or low salinity systems, in which we can also, for example, find these hydrocarbon reserves.
Jadi untuk palynologi ini, kalau kita punya data polen, kita juga bisa menggunakannya untuk mencari sumber gas dan sumber minyak seperti itu ya. Jadi kita bisa mengetahui nih pemetaan minyak itu ada banyak di bagian mana, itu kita bisa dengan mengetahui komposisi polen di satu daerah itu. Jadi kita bisa mengetahui dari jenis polen nih, oh ini polennya pasti dari daerah terestrial atau daratan gitu. Atau kita menemukan polen bentuk lainnya, oh ini pasti daerah polen-polen seperti ini nih, atau kondisi seperti ini menunjukkan bahwa daerah ini kondisi garamnya itu rendah atau kondisi garamnya tinggi seperti itu. Jadi kita bisa menggunakan palynologi untuk memetakan kondisi lingkungan di masa lalu sehingga kita tahu sumber minyak itu ada di mana.
Yes, then one field that I'm always quite intrigued with is forensics and criminology, where we can use pollen as well. So actually, forensic Palynology majorly uses pollen grains to support crime solving by specifying where a subject or an object has actually come from after or has been after the pollen has been collected from cloth or, for example, shoe ridges, as you can see in the picture below, or just the skin. So we can sample pollen from there. And the pollen composition then actually gives us an indication about the region, because every region, every location has a very unique and specific pollen composition simply by the plants that occur there, and also by the means of disposition. And this can uncover actually, the origin of the suspect or object, and even sometimes the season in which an object has been exposed, because, for example, in Europe, plants flower only in spring and summer, and in winter, for example, they don't flower anymore. So we know when we find a lot of pollen on the clothes that it had to be there in spring or summer. And yeah, maybe you also know the series Sherlock from BBC. I don't know, maybe some of you. And here actually there has been one episode in which Sherlock Holmes used also pollen from a shoe to identify a culprit or to know where the culprit came from.
I think our student know. Jadi di sini untuk palynologi ini bisa untuk mengidentifikasi kasus forensik dan kriminal ya, terutama kita menunjukkan dari mana asal si subjek, baik yang pelaku kriminal atau yang korbannya seperti itu. Kita bisa melihat nih di filmnya Sherlock Holmes ini, jadi dia pernah ngambil sampel dari tanah yang ada di sepatunya korban, itu mengetahui bahwa korban ini dipindahkan atau seperti apa gitu ya. Jadi ternyata si film Sherlock Holmes sendiri udah pakai konsep palynologi di sini seperti itu.
One example also maybe is how pollen has been used to trace activity in illegal mass graves in Bosnia. And here actually the people, they were buried separately, but when they were buried in like a mass grave, this mass grave was made in a wheat field. So actually on the bodies there was pollen from wheat. So they found like a lot of pollen from wheat then on these bodies. And then secondarily, they have been buried separately to like cover up that they had a mass grave before. They pretended that they actually buried them separately, but they could find the pollen from the wheat still on the clothes. And they knew that the location of burial before must have been at a different place, and actually all in the same area. And another example maybe is just a burglar who has been catched because there was pollen of Hypericum on his clothes, and around his area of living there was no Hypericum, and the place where the burglary has been conducted was full of Hypericum bushes, and well, then they could find out that he was there. Yeah, just some examples here maybe.
Jadi ada beberapa contoh nih kasusnya. Yang pertama itu misalnya ada kuburan massal ya. Jadi kuburan massal yang bisa jadi itu kuburan massal yang ditemukan polen di pembunuhan massal yang ditemukan polen di tubuhnya. Jadi walaupun dia dikubur terpisah, tapi jenis polennya sama yang ditemukan pada setiap badan dari korban-korban, itu menunjukkan bahwa sumber dari korban-korban yang dibunuh ini ternyata sama, seperti itu, satu tempat gitu. Atau misalnya ada penyelundupan. Jadi kalau misalnya ada penyelundupan itu ditemukan di bajunya itu ada polen tertentu, ternyata polen itu tidak berasal di daerah di mana dia menyelundupkan barang itu, tapi malah di daerah lain, itu bakal ketahuan dia menyelundupkannya itu dari daerah mana. Seperti itu.
Okay, thank you. Yeah, so let's get to biology just quickly still. So of course, pollen can also be used as a pollen forecast, actually, for major allergic pollen groups. So I have a pollen allergy myself, and this is quite helpful because we can actually trap pollen in the air and find out when we have a high pollen content of certain groups. For example, I don't know, certain taxon like hazel, and then you can avoid going outside basically, so that you don't have such a strong allergy attack for hay fever. This is done already weekly all around Europe and also the US. I don't know how it is in Indonesia, if you have a bit of a weather forecast here. But we can, for example, also find like regional pollen calendars where we know when exactly in which month we have a high content of this certain pollen in the air.
I don't find it in Indonesia forecast.
Sad. Yeah. Okay.
Maybe because many pollen every season.
Yeah, maybe it's throughout the whole year. That's true. Yeah.
Jadi di sini kita juga bisa mempelajari polen itu untuk melihat ramalan cuaca ya, ramalan cuaca polen gitu. Jadi kalau di Eropa itu sudah ada diketahui kapan polen itu tersebar gitu. Jadi ramalan cuacanya, apalagi untuk orang-orang yang alergi terhadap serbuk sari ya. Jadi ada informasi di perkiraan cuacanya ini di daerah ini banyak polennya, tingkatan polennya tinggi seperti itu. Jadi kita akan menghindari untuk keluar rumah atau pergi ke daerah tersebut gitu. Jadi di Eropa itu sudah umum gitu ya. Kalau di Indonesia kita mungkin belum ketemu atau kita belum ngeh kalau kita ada yang alergi polen. Okay.
Yeah, and last but not least, pollen can also be used for food security. Actually, here studying honey called Melissa Palynology. Yeah, which here again a Greek word Melissa, which means bee. And the analysis of pollen and honeydew elements is meant by that in honey. And we can study here, for example, the geographical origin of unfiltered honey by the composition of pollen that we find in the honey. So when we find, for example, a high amount of one taxon, we know that the bee must have visited this taxon and this maybe just grows in a certain area.
Jadi ada lagi ilmu tentang palynologi ini berhubungan dengan palynologi yang berasal dari kata melissa, itu artinya adalah lebah ya. Jadi ingin mengetahui tentang madu seperti itu ya. Jadi kita bisa menganalisis ini nih si polinator atau si lebah ini dia mengkonsumsi madu di daerah mana gitu. Karena kita bisa melihat atau kita bisa melihat keragaman dari madunya itu seperti apa yang dikonsumsi oleh lebah dari polen yang kita temukan di madu.
And of course, maybe a little bit of part of this is also plant-pollinator interactions and studying pollen loads from pollinator organisms. Majorly, this has a scientific background, so we actually want to find out where these pollinator organisms have been and which plant sources they use for their production of nectar or honey, or also simply for food. And this is, of course, important to understand how our ecosystem functions, as pollinators are certainly a key group and produce actually, like, for example, just almost all of our food actually. So we should actually know about how pollinators interact with plants in our environment. For example, here, bees or bats. And this can be done in many different ways. So just an example that we use in our project is how experimental stingless bees visit like different areas. So we have these beehives, but also you can do it with sweet netting or simply studying the pollen load from manually catched bees. Yes.
Jadi di sini kita juga bisa melihat interaksi antara polinator atau penyerbuk dengan tanaman ya. Polinator itu bisa beragam, ada lebah misalnya, ada kelelawar ya. Kita bisa mengetahui juga ini ada bagusnya juga untuk mengetahui ini asal tanaman kita itu dari mana seperti itu. Jadi kita bisa lihat misalnya hubungannya antara lebah itu dia konsumsi pakannya itu dari jenis-jenis apa saja seperti itu. Demikian juga dengan lebah. Jadi kita tahu misalnya kita nih punya tanaman tertentu, kita tahu penyerbuknya itu juga dari mana gitu. Dengan mengetahui polen-polen yang ada di sekitar kita. Ini biasanya sih bisa dilihat dari bisa jadi polen trap atau bisa dari hewan polinator itu yang kita cek polen yang ada di tubuhnya itu apa saja.
Mm-hmm. Yeah, so actually this was it from my side. So thank you, terima kasih for your patience. I know this was quite long, I think majorly also because of the translations. But thank you for the effort, Ibu Hesti, to also communicate everything to your students. And maybe there's still some questions, or maybe it's too late for you guys already, I don't know. Yeah, but thank you very much.
It's not too long, but because it is interesting, I think it's not any such effort to communicate with student. Oke, siapa yang ada mau bertanya, ya silahkan ditanyakan, boleh via udah banyak pertanyaan. There is some question in the chat. Jadi very many questions. Oh my god, I will choose some of them. I will read for the first. I will read first, and then we allow the someone that raised the hand. First is from Rika Kitri Ardianti. She asked, she asked, how we said before that pollen we can found in the tips of Netherlander, and then how it's be fossil and it's still keep in the teeth of that humans.
So basically, they are catched in the crevices of the teeth. So the teeth, they are, they have a lot of ridges. And the pollen will stick inside of the ridges for a very long time, actually. It's quite amazing. But the pollen is so small and it's so sticky that it just stays. Yeah.
Oh, yes, amazing. Jadi kenapa kok bisa ditemukan pollen itu di gigi? Ya, jadi yang ditemukan itu di lubang gigi dari manusia Neanderthal tadi, dan dia itu tersimpan atau terjaga di sana karena setelah polennya tersimpan, dia kan lengket ya sifatnya. Kemudian ada kayak karang gigi yang menutupi polen itu sehingga dia masih tertahan atau terawetkan di sana seperti itu. Jadi itu kenapa bisa ditemukan si polen di gigi manusia purba tadi. Next we allow Nanda to ask the question, please, Nanda.
Yes.
Can unmute first. Bisa di unmute dulu, Nanda.
Okay. Thank you, Ms. Hesti, and thank you for the opportunity. And first, maybe I will thank to Ms. Pea for the great presentation. And very interesting information, I think, that how palynology is very useful in various fields of life, like in forensic and aerobiology as on. And to be honest, this is something we have all just come to know in a greater detail, I think. And based on that, your presentation before that, research has been carried out in several areas or regions, right? And what are the differences in each pollen in that area? Perhaps from a morphological point of view, maybe, and what causes the different characteristic or traits of each pollen? That's
So,
all the question. Thank you.
yeah, thank you. Thank you for the question. So you
Yeah.
mean the morphological difference in one genus, in one species, in different regions? Or you mean the general morphological difference of different taxa and different genera?
Yeah, maybe in the from the differences in each pollen in that area that you did in your research. Yeah.
So basically, every plant taxon, every actually species, but yeah, morphologically it's sometimes hard to find out or distinguish without electron microscope. Each genus or each species has a unique pollen grain. So every pollen grain from one taxon looks different. However, one taxon produces the same pollen grain. So when you look at regional differences, for example, when we look at the tropics, we find pollen from tropical plants. So all the tropical plants, they produce pollen, and this pollen can look morphologically different to plants that we, for example, find in Europe. However, this is not correlated to the region, it's correlated to the plant taxon. So basically every plant produces a different type of pollen. What is interesting, however, is that the same species can regionally produce pollen that maybe has a slightly different size. So sometimes it happens that, for example, in the high north, we have pollen grains that are a little smaller than in the south. But this, I think, is mostly correlated to environmental conditions. Yes. Does this answer your question?
Yeah, I think I don't need to translate to you, Nanda.
Yeah, English is good.
Hearing this is already good.
Mhm.
Nanda, Nanda have good sign also.
Oh, yes.
Yeah.
Okay, thank you for the explanation, Ms. Pea. I do understand what your the explanation. Thank you. Danke.
Kein Problem.
Nanda is one of lecturer in our department. Yes. She
nice.
also teach. Yeah, she also teach English for biology.
That's
her
good.
English is good, I think. Okay, we continue to the next question. It's from Shifa Nur Rahman. She asked about the pollen size. Is the pollen size fossil pollen different? How about pollen fossil? It is the size different with the modern fossil. Yeah, how could be done?
So in general, the pollen grains that you find fossilized is from the same, usually the same taxa that you also find nowadays. So originally the size must have been the same. However, when it undergoes fossilization, so actually it loses, for example, the insides, and there's no like organic compounds, but the sporopollenin left, it can shrink. Especially when we use some chemical treatment, the water goes out, everything like dries up a little. So they look smaller than when you have the fresh pollen. So the size will change, but not because it's like an actual morphological change. It's just because of the treatment or because of the conditions the pollen has been preserved in.
Yeah, Ya, jadi Shiva, polen itu masa lalu dan masa sekarang kalau dia berasal dari tumbuhan yang sama, itu bentuknya bakal sama. Tapi kalau ukurannya itu bisa dia menyusut yang untuk polen fosil karena kondisi lingkungan. Tapi secara basic bentuknya itu sama, ukurannya itu menyusut karena kondisi, baik itu kondisi lingkungan atau stres atau tekanan yang mengenai si polen itu, seperti itu. Actually, Shiva has two questions, but the second question is the same with Nanda. Okay,
Nice.
okay.
Yeah, it's a good
Someone
question.
raise hand? I don't see. Ada yang raise hand? Ada yang mau tunjuk tangan? Oh, nggak ada lagi ya. Okay. Okay, we continue to the next question. It's very much. It's difficult to to
No.
yes. Nah, itu ini. Ya, Nilam. Okay. From Nilam. Nilam asked, as when you conduct the palynology research, there is some obstacle to find the pollen fossil, and what kind of the newest pollen that you have found?
So you mean like when I study fossil pollen?
Yeah,
which is like the newest one that I find, or?
Yeah.
Okay. So basically we study pollen from sometimes just 10 years ago. So it actually depends on like when I take a sediment core, like let's say I take a sediment profile from an environmental archive where pollen has been preserved in because of, for example, anaerobic conditions. I take samples throughout the entire core. That means also the part of the core which is young. So also the upper part of the core which only recently accumulated. So this can be pollen from just a few years ago up to thousands of years ago, throughout all the time range. It can be any time you want, basically, until like a certain limitation, of course, where pollen is not preserved anymore. But yeah.
Do you find some obstacle when you conduct the research in palynology?
Yes, there's a lot of obstacles. So on the one hand, of course, identification. So identification you saw is sometimes really, really tough. So we have a lot of different morphotypes, which we don't always know. So we actually don't have so many studies on different species and their pollen types in the present. So sometimes it's almost impossible to find out where the pollen grain comes from that you are studying. And of course, the level of preservation can be really hard. So sometimes the pollen is preserved really badly, and then the pollen grains break apart, certain features are not visible anymore, maybe the wall is not so nice, and then you also cannot identify it anymore. So this is like some challenges we are facing.
I have tried and it's very difficult, I think.
Yes.
Jadi, kendala dalam penelitian palynologi ini ada beberapa ya. Ada yang pertama itu kadang kita menemukan polen itu yang susah sekali diidentifikasi gitu. Bisa jadi karena bentuknya yang sangat tidak biasa gitu. Jadi Spriani udah melakukan penelitian selama 10 tahun ya untuk palynologi ini, dan kadang dia menemukan untuk polen itu sumbernya itu sumbernya itu nggak tahu mau dirujuk kemana, atau juga menemukan ada yang polen itu dinding selnya sudah rusak ya. Jadi ada yang pada saat persiapan atau perlakuan di lab itu malah nggak ketemu itu si polen ini jenisnya apa, karena bisa jadi rusak karena ada kesalahan pas perlakuan gitu, macam-macam. Jadi ada banyak sekali tentunya permasalahannya. Kemudian tadi pertanyaannya apa tadi ya? Satunya lagi polen terbaru. Oh ya, kalau polen itu kalau kita ngambil polen itu kan kita ngekor gitu ya. Kita ambil dalam tanah itu kita akan melihat itu susunannya. Jadi kalau kita ngambil polen itu kita nggak boleh tanahnya itu nggak boleh diaduk-aduk, jadi harus deretan. Jadi polen terbaru yang kita lihat itu adalah polen yang paling atas. Jadi polen yang paling tua itu usianya itu paling bawah. Jadi ini banyak sekali menemukan polen-polen yang zaman sekarang. Jadi polen yang terbaru ini yang ditangkap itu adalah polen yang di modern sekarang berdasarkan core-nya. Do you have a new species there in the pollen identification?
Sometimes you find pollen which you don't know where it comes from. So this is then like a new type that maybe also hasn't been registered before. Of course, it can also happen that you maybe in the past find types from species that don't even exist anymore nowadays. But of course, you don't know then where it comes from, because you need to first have the plants, look at the pollen from the plant to then be able to trace back the pollen to this plant. So we need to study the present pollen to indicate the past pollen. Yeah.
Jadi saya juga menemukan beberapa polen itu yang tidak ada cocok lagi dengan polen tanaman sekarang. Bisa jadi polen-polen itu tidak ada di zaman sekarang atau sudah punah seperti itu. Jadi mungkin kalau tadi nanyanya apakah polen jenis terbaru, itu mungkin nggak ada ya, karena polen itu sudah ada dari dulu, dan kalaupun tidak ditemukan sekarang itu berarti bisa jadi dia telah punah, karena tidak bisa dicocokkan lagi gitu ya, atau kita tidak tahu ini morfologi polen ini merujuk ke tanaman apa. Okay, there is very much question, but I think because yes,
maybe not all.
The student is very enthusiastic, but I think we also here also our lecturer named Ibu Tri Susanti. She also I just know she also conduct the research about pollen, and she she used a microscope electron to identify the pollen, modern pollen, I think. I didn't see Ibu yet.
For taxonomy, probably.
Yes, for taxonomy. I think we should finish our meetings. Yeah, it's very happy to meet you, and
of course.
having you here.
I
hope we have another time to speak about maybe palynology and other subjects, something like that. But before our finish the lecture, maybe you have some note or some motivation that you want to say to our students.
I always, of course, first of all, thank you, Hesti, for organizing this. It was a pleasure for me to also speak, because it's always very dear to me to actually engage students into palynology and also make this topic available, because I feel it's often quite unknown how many possibilities you have with studying pollen, and to also encourage you actually to work in palynology. So of course it's not easy, but especially when you enjoy morphology and you enjoy identifying, this is like a playground for it. So you have a lot of possibilities, and especially you fit your interest. So there's so many different fields. You will find something that you're interested in working with palynology, and I always hope to gather more people to join. So don't be hesitant to try it out, and maybe also for your final thesis or something, just try to find topics and to engage.
Jadi pesan saya terakhir, kalau misalnya kita tertarik dengan morfologi, polen ini merupakan suatu hal yang baru yang masih perlu digali lagi. Jadi ada banyak banyak hal-hal menarik yang kita bisa pelajari di polen ini. Jadi jangan sungkan untuk melihat tentang mencari tahu lagi tentang polen ini, seperti itu. Nanti mungkin yang lain kalau misalnya belum puas tentang apa yang disampaikan saya, atau misalnya masih ada yang bingung dengan terjemahan saya, mungkin yang tidak 100% bisa dilihat lagi di recordingnya. Saya juga udah share di YouTube, bisa disimpan tuh link YouTube-nya buat dilihat-lihat lagi, seperti itu. Okay, thank you, Svea.
Yeah, cheers.
Yeah, thank you for coming here and give the new information for our student, because when I study palynology for first time with you and Chung, and I think it is actually it's not new insight, but it's new for me. So I think because of that, my student also feel the same and they have many questions. Could I share your email to if they have any question?
Yeah,
Okay.
certainly. It's no
Yeah.
problem. Yeah.
Okay. Nanti kalau ada yang mau tanya-tanya tentang palynologi, saya akan share emailnya Svea ke kalian. Kalian boleh minta ke saya, atau nanti kalau ada yang mau di kelas, saya akan kasih di grup seperti itu ya. Okay, Svea, thank you so much for coming here, and I hope everyone get the good insight here. And good morning there.
Yeah. Yeah, of course. Yeah, thanks a lot. Yeah, for this and for your hosting and everyone listening. Okay.
And
then,
and say hi to Chung.
Yeah, I will.
When you go to office,
when I can go back and I don't no longer positive.
Yeah,
of course.
I hope you get well soon and not positive anymore.
Yeah, I hope so too. Okay, yeah, thank you all. And cheers. Bye-bye.
Bye. Oh yeah, maybe we need to take a picture together. Oh yeah. Sorry, sebenarnya udah keluar ya. Telat ngomongnya ya. Oke, sampai di sini mungkin semuanya, karena udah masuk maghrib juga, kita sudahi pertemuan kita sampai di sini. Ya, mungkin kita perlu foto bersama dulu buat absen, ya, buat Ibu Fevi dan Bu Tri, dan juga yang mengambil mata kuliah pilihan. Oke, siap Bu Fevi? Ya, silakan semuanya. Kameranya di on kan ya?
Oke, saya mulai dari slide yang pertama ya. 1, 2, 3. Slide kedua. 1, 2, 3. Slide yang ketiga, yang terakhir. 1, 2, 3.